video on, we're going to see you in the video if you pop up or ask a question. But if you don't want to be in that position, you're gonna have to leave the meeting and go and watch it later. Can somebody type a question quick actually for me in the chat, just so I can see if that's gonna show up for me while I'm doing my screen presenting? I should have just assigned somebody to do it. There we go, Grace, thank you. All right, so I can still see those. All right, so let's go into Schoology, which is always unavailable these days. We should graph the average number of times you have to refresh Schoology before you can see the page. You'll see 3 new activities. Well, you may only see 2 new activities. They're at the bottom here that I've added. Well, I opened up this morning. We're going to go through this analyzing temperature's effect on enzyme activity page. So you'll see it on my page, so you can go back to this later if you want. But this is going to be your model for what you're going to do. And then you have been already assigned one of 2 different activities to analyze the effect of either pH or substrate concentration on enzyme activity. So if you've done Monday's assignment, you should have enough background understanding to do this assignment. If you've completed the notes already, which aren't due until Friday, you'll already kind of know what to expect. But this is going to just be an introduction into how to use models to simulate research. So this is the document that I have linked on that page. This is essentially the one I'm going to do with you, and then you're going to be doing something similar, but with a different variable. So enzyme activity, the ability of an enzyme to carry out a reaction, to facilitate a reaction, is affected by environmental conditions, right? So the rate at which a reaction will occur in the presence of that enzyme can be affected by things like temperature and pH and so on. So this lab is about doing an experiment on that. All of you are going to have the same background questions to answer first about the lactase enzyme, which is the enzyme that we're going to be studying. Now, one of the things that's confusing about lactase, or not confusing, but it's easily mixed up, is that lactase is the enzyme. The thing that it's breaking down is lactose. Lactose is a carbohydrate, a sugar that's found in milk, and its lactose is broken down into 2 other kind of sugars, which is galactose and glucose, which is the sugar that we can most easily use. But we can actually use galactose as well. So the enzyme's job is to break down lactose sugar. So be careful when you see or read the words lactase and lactose that you think, all right, am I talking about the protein or am I talking about the sugar? But you're going to do a little background reading. It's pretty simple. Answer a couple of questions. I see I misspelled the word catalyzed here. And then you're going to have a simulation. And the simulation is going to come from this website here. So yep, that's what I want. So this is what your simulation is going to look like. And basically this is just a digital tool that we're going to be using a lot in the next few weeks, where you can design and run experiments to learn things about all kinds of stuff. If you look at the top, we'll be able to learn things about metabolism, or we could do genetics or ecology or whatever. And it's a way to bring experimentation and data manipulation into a digital world when we can't physically. If we were in class, we would be doing these experiments in class with actual, test tubes and lactose and lactase, and we would be boiling it and doing all that stuff, but we can't do that digitally. But this recreates it. So in the lab, it tells you first your design. So it says start with the initial lactose at 48 millimoles and the pH at 7. It says don't vary these parameters while testing for temperature. They are the environmental controls. So I set that up, okay? And so if I go back here, I want 48 and 7. So here's my lactose in millimoles. Look at that. It's already at 48. I want the pH at 7. It's already at 7. Now, if I needed to change that value, I can just slide these around and it'll tell me where I'm at, and I can just set it there. Now, I realize that as I'm doing this, I can't see if people are asking questions. Nobody's asking questions. All right, so I'm going to assume that I'm still good. So I'm not going to vary those while I am running the experiment, because what I want to test is the effect of temperature. So if I look at the lab, it says set the sample size to 5, which will cause the simulation to run 5 samples. This would be like if you had 5 different test tubes and you were running the same experiment 5 times. The nice thing about this digital simulation is it'll just do it 5 times for you, and you can see the variation. Now, part of the reason I really like this simulation is they have it look like a real world experiment. So in the real world, even if you do everything exactly the same, you have the temperature and the pH and the lactose and everything the same, there's always variation. With enzymes, the collision theory says reactions occur when things run into each other, and that's still a random process. We can predict things about it, but it's random. So if I run this, you're going to see 5 lines appear on the graph, which represent 5 different trials. And you'll notice there's some variation, but they all follow the same kind of trend. If I was to run it again, I would get 5 other lines, which aren't exactly the same as the first 5. So there's variation, right? Which is great because that's real world. So it says I'm going to set the sample size to 5, which I've done. And then I'm going to vary the temperature by 10 degrees Celsius, recording the rate of reaction per minute over the first 10 minutes in the table below. Okay, so I have my temperatures up here, and I have my 5 reactions. Now, I've already run this for 0, 10, 20, 30 degrees Celsius. I'm going to run it for 40 right now. So I had set my temperature value at 0. I ran it, right? I recorded that data. I went to 20 and so on. Okay, so I'm going to set it at 40 degrees Celsius, and I'm going to run it, and I get my data. Now, what I'm supposed to record is the first, second, third, fourth, and fifth reaction, what the reaction rate was for the first 10 minutes. So I'm going to need to do a little math. And the easiest way to do that is to go back to your graph. If I put my cursor on the graph at 10 minutes and I click and hold, it's going to draw a vertical line here showing me what's happening. And you'll notice underneath the graph, right underneath the graph, it says time in minutes is 10. And then it gives you sample 1, 2, 3, 4, 5, and it gives you the glucose values after 10 minutes in millimoles. But that's how much glucose there is after 10 minutes, and I want to know the per minute rate of reaction. So the math's easy. I just divide that number by 10, and that will tell me on average over the first 10 minutes how much lactose was getting turned into glucose by the enzyme lactase, right? So it's the reaction rate. Okay, so for my first value at a temperature of 40 degrees, I have 5.29 millimoles of glucose made. So lactose turned into glucose. If I divide that by 10, my first value that I would put in here is 0.529. Okay. All right, I'm going to go back into my meet here and see if there's any questions. Does anybody have any questions they want to type out right now? We good? Okay. So I'm going to get the rest of my values here. I'm going to just write them down quick, and then I will transfer them all over. So at 10 minutes, I'm going to have sample 2, and you can follow along with me, is 0.594. Sample 3 is 0.474. Sample 4 is 0.509. Sample 6 is also 0.509. All right. So now I'm going to go back here and I'm going to put it in my data table. Whoops. Well, I did it out of order. Okay. So I have all of that data now, right? And this will have taken you longer because you'll have had to start at 0. I did all this yesterday just to speed things up. And you'd record, you'd run it at 0 degrees. And then you'd look at 10 minutes and you'd realize, oh, the reaction rate for all of them is 0. And then you'd run it at 10 and so on, right? And then you'd realize, oh, the reaction rate at 10 degrees. You record those values and so on. Okay. And you'll have all your data recorded on here. But now we need to do some data analysis. And this is where I'm going to show you how to switch over and use a Google spreadsheet. So I'm going to copy this data. If you were doing this, you would copy all of the data. Okay. But I'm just going to copy this data. So I have the other already. And I'm going to go into a spreadsheet, which already is set up with all of the data from earlier. Put that in there. Now I'm going to show you how to do some of the data manipulation. And again, I've already done some of it. I'm just going to show you 2 of them to show you the process. Okay. Oops. I see I put those into the wrong place there. There we go. Okay, so first we need to find the mean reaction rate or the average. So to do that, you click on the cell where you want to put the average. So I need to get the average at 40 degrees Celsius, and I type equals, and then AVG. Okay, and then I highlight, and it's going to give me a parentheses, and I highlight the cells that I want to average. And then I close the parentheses and I hit enter. Oops, what did I do wrong? Oh, I think I did the wrong average. Equals average. Oh, there we go. I think I had rank average. I don't know what that is. I thought it was going to work the same. It obviously doesn't. There we go. So there's the average of all of those data points is 0.523. Okay. Then I need the standard deviation. Well, how do you calculate standard deviation? I'm going to do this twice so you can see it happening twice. At 30 degrees Celsius, I'd already calculated my average. For this, I just put equals STD, right? And then it'll pop up standard deviation. Okay. And then I have to say which cells I want the standard deviation of. I do not want the mean as part of this, so I need to make sure I'm only including the sample data, the first 5. Close the parentheses. There's your standard deviation. We're going to do the same thing here. Okay, and now some of you may have noticed that it was kind of guessing what I was going to do. If it guesses correctly, you can just hit that, but I'm doing it kind of a long way. All right, so now I have my standard deviation for all of them, and I need to calculate my standard error of the mean. Let's go back to the lab to see why that is. Because in the data analysis, what you're going to be asked to do is to create a labeled graph to illustrate the sample means. I'm going to come into the meet here and see if anyone's got a question. All right, so where did I get the 0.529? Oh, we figured that out. So for every trial we run, okay. So Jermaine's question is a good one. Does lactose change during the test? Yes, it's wait, hold on. Let me go back and make sure I'm in. Does the lactose change? Okay. Yes, the lactose changes. I don't actually change these values because each time I run the trial, I want to start with the same amount of lactose. But in the reaction, the lactose is turning into glucose. That's the chemical reaction. So yes, it's going to change. When we graph it and we put our line here at 10 minutes, at the bottom of this page, all of these values, 13.5, 17.4, that's how much glucose was made in the first 10 minutes. So I think I'm answering your question here, but that's where you get your 5 samples. There's going to be others, sample 6, 7, 8, 9, but those just didn't get run. You can do this up to 50 samples, right? So if you wanted to, you could run a ton of samples, but we don't need that much data. 5 samples is enough. Okay. If I'm not answering that question, how many decimal places do you want us to go out to? Just use as many as they give you. It's going to be like 3. The way the model is set up, the simulation, it's that precise, so we can be that precise. If we were doing this in the lab, we wouldn't have that precise of data, but there is lab equipment that can do it that well. Any other questions right now before I get into how to do some of the more of the data manipulation? Okay. I'll come back to that page in just a second, so if there's more questions. Okay, so standard error of the mean. The equation for standard error of the mean is the standard deviation divided by the square root of the number of samples. So it would be the standard deviation divided by the square root of 5, because we took 5 samples. So there's unfortunately not a simple equation to just type in standard error of the mean. So to do that, I put in equals, and then I will do a parentheses, and I will click this box because I want to take the mean, standard error of the mean, or I want to take the standard deviation, and then divided by, and then I want to take the square root. And so you can type in SQ, and you'll get squirt, which is the square root of, and it's going to be 5. I'm just going to type that in. I'm not going to highlight anything to do that because I know there were 5 samples. If I hit enter, that'll give me that standard error of the mean. I'm going to do the same thing here. So equals parentheses. Now I need to make sure I click the standard deviation from the 40 degrees Celsius data. Okay. Divided by SQRT of 5, square root of 5, because there were 5 samples there, and I hit enter, and there's my standard error of the mean. Now there are some shortcuts that are speeded up a little bit. It doesn't speed it up that much. If you know what they are, use them. If not, don't worry about it. Okay, now standard error of the mean is only one value of standard error of the mean. So if we look at the lab, it's going to say create an appropriately labeled graph to illustrate the sample means at the 5 temperatures tested to within 95% confidence, which is the mean plus or minus 2 standard error of the means. So let's quick check and see if there's any questions. Okay, so I can at this point go back in here and do 2 standard error of the means, which that's just going to be 0. This one I can do equals 2 times the standard error of the mean. Oops. Equals 2 times, let's see if it'll work this way. Oh, it's not going to let me do it. All right, I would just do the math then on my own. It'll just be easier. Sometimes I just go old school. Now, Riley, your question comes into play. I just put, 4 or 5 of those digits after the decimal point in there. So 2.01692 or so. And I'm going to multiply that times 2. And I will get 4.033. Okay, 8. Great. All right. So that's my standard error of the mean for that. 1.47. I should have done this ahead of time. 6.2. 2.95. There is definitely a way to do this on Google Sheets. I just don't know what it is. If you want to take the time to figure it out, go ahead. Okay, that'll be enough. 2 standard errors, the means will work. Okay, you would do this for all of them. Okay, no questions yet. All right, so back into the spreadsheet. Now I need to make a graph of the means. So I would start by highlighting the mean values, okay, from the mean of the reaction rate. I'm going to copy those, and I'm going to bring them down here into another section of the table, just so that I can work with it in graph form. Now I'm going to highlight this area. Wait, why is it not? Oh, it's putting the function in. There we go. Okay, so there are my means. So if I highlight all of those, and then I go up here to insert chart, there's my chart, along with my values at, that does not make sense. Hold on. What's going on here? Why is that? Oh, what? I see what's wrong here. Ha! I didn't put these in. All right. Forget that. I'm going to go back. I got to put these values in again. If you guys noticed, here I have them in decimal point and, or I have them as a rate per 10 minutes, but here I do not. There we go. No, why is that still not? Okay, hold on. Come on, data. Come on over. That's not working. I don't want to do it that way. I'm sorry, guys. Should have tried this out beforehand. Try it up here. There we go. So this is 0 Celsius. I got to get my, well, this one's just going to be 0 equals AVE. Now why am I? Sorry, guys. This is what happens when that's done. Okay. So now I've got my averages, and they make more sense. Oh, I gosh, why is this not working? Okay. 1.336. 0.523. Okay. All right. Now we got our data. And you'll see these updated as well, the standard deviations and the standard error of the means. You guys got to watch your teacher struggle a little bit. Are there any questions before I'm ready to go back on here? Let's see, it looks like there's one. Did you calculate... How did you calculate 2 standard error of the means? I took the standard error of the mean and then I just multiplied it by 2. I hope that makes sense. All right, good. Okay. Yeah, I don't know why it wasn't working just typing in average. It did earlier. I think if I hadn't, I apparently had copied the data over incorrectly initially and it set something else up weird on the thing, but I think we're good now. Okay, so I want to graph this data. So let's go back into insert chart here. And that, what? Why is this looking? Delete that chart. Yeah. Why is it graphing the okay,
wait, Burns, I think make a graph. If you make a graph and you go below, it says like use like columns or something as headers. So if you make it a graph, and you like unclick one of the boxes, I think it might work.
Okay, hold on. Let me try this and then I'll try that, Grace. I think that was Grace, right?
Yeah.
All right. I think I think that way does work also. Let me just, since I was already in here, let me just try this here. Yeah, you're right. Okay, so no. Use row 19. No, that's not what I want. Switch rows and columns. Why is it not doing that? All right, what am I doing wrong here, Grace? This worked earlier. I don't know why it's not working now.
If you go to your one with the 0 to Celsius and then the 0, if you highlight that.
Okay.
And then make a chart. The other data.
Oh, okay. This data up here?
Yeah.
Okay, make a chart of this. And then
it says use row 15 as labels. I think you want to unmark that. Or then do the switch rows and columns.
Why is it putting them all as 0? Oh, here, I think I know why. Let's do this. Let's try this. Maybe I know why. Either way, once this is... All right. Why are these values still... Okay. All right, one last thing. Hold on. What if I just get rid of these entirely? No. I don't whole new sheet. That's why it's putting it in as reference data. Okay. All right, let's try graphing that. Still showing up as don't want to use column A as headers. Okay, you know what? Here's what I'm going to do, because I don't need you guys to sit here and watch me fumble. I know I had this working yesterday. I'm going to go as soon as I get off this call, and I'm going to create a separate video of how to just do that part, and I'll just explain to you the last bit of the lab, and then you can go back and watch that video of how to do the graph. And hopefully on that, I'll explain how to do that. I don't understand why it's not working for me today. Either way, once I make that graph and I've added the 2 standard error of the means, which I would have calculated, which I did calculate here, okay, and I will show you in that graph how to, or in the other video, how to do that. Once you've done that and you've added the error bars, you're going to, I'll show you how to download that graph, and then you're going to copy it into here. So that graph will go there. And then you're going to answer some analysis questions. What were the results of the simulation? What was the optimal temperature? And I'll do that for you in here. Question number 4 is the only one that could be a little bit tricky. It's going to ask you to do some background research on where lactase is found in living things, and then relate the activity observed in the experiment to the location of the enzyme. When you do that, you're gonna, and the reason you're doing that, and I'm not gonna give this away because you're all gonna be doing this in your own experiment anyway for lactase. Enzymes have evolved to work in very specific parts of the body, so or of living things, and because of that, they function in a narrow environmental range because they have evolved to do that. So what that means is if you have an enzyme that's in a part of your body that's highly acidic, it's going to be optimized to work in an acidic environment. If it's in an environment that's not acidic, it's, you know, that's neutral, it's going to be optimized in a neutral environment. If it's an enzyme that's designed, part of the, not designed, that's not how evolution works. If it's in the body of a living thing that's found in very hot environments, the enzyme will work best in hot environments, cold environments, and so on. So you're going to read a little bit about lactase, and then you're going to try to connect why it works the way it does. Now, if I had finished the graph today, you would have seen that lactase is optimized to work between 25 and 30 degrees Celsius, which is exactly the human body temperature, which makes sense. So the enzyme works best in that environment, meaning its shape is designed to carry out chemical reactions at that temperature. And if it was going to carry out chemical reactions in something which was colder or warmer, it would be, its shape would be different so that it could carry out those reactions at different temperatures. Your task now is to go and do a similar assignment, but instead of looking at the effect of temperature on enzyme activity, you're going to be looking at the effect of pH or of substrate concentration. And I've assigned, I don't know, I just kind of randomly assigned people to each of those groups. But when you open that, so for instance, if you're doing the pH one, of course, Schoology is not going to be open right now. If you opened up the pH one, it will look just like this. The only difference is you're going to have to design the experiment, meaning you're going to have to tell me how to set the parameters initially. So I was manipulating, my independent variable was temperature. So my initial environmental situation was the concentration of lactose and the pH. I wanted to hold those steady. Those were my controlled variables, right? I had the sample size set at 5. That was the number of times I was going to run it. And then I had to describe what was my independent variable, temperature, and I was going to adjust it by 10 degrees Celsius. And then my dependent variable, which is the reaction rate per minute over the first 10 minutes. You're going to have to design your own experiment using this website. So if you're supposed to investigate the effect of pH, you're going to need to figure out how you will control the other variables you can control. You're going to need to decide how you're going to vary pH and what your dependent variable is going to be. What is the thing you're going to count? Hint, hint, you should count the rate of glucose production, right, over the first 10 minutes. But you're going to write that out because I want you to see how to design it. Now, as far as what it'll actually look like, it's going to look like a lot like my design here, just set up to test pH or set up to test concentration. You're going to have a data table. It's going to look a lot like this. You'll need to put the data into here and then bring it over into a spreadsheet. Okay, you can let the spreadsheet do your standard deviation and standard error of the mean calculations for you. Based upon my problems today, I would say calculate the mean on your own and just input it as a number, although you may not have to. And then you'll have to calculate the 2 standard error of the means by doubling those numbers. You'll graph it, which I'll have a video for you in a bit. You'll add the graph in and you'll answer some analysis questions. Are there any questions about that lab right now? Okay, are there any other questions about other stuff that you guys are running into for the class or for your other classes or about online learning overall?
I did have a question. So for the like statistical tests and stuff, and then the notes that we were supposed to take on Monday, you said that was due on Friday, or was it just due on Monday then?
So the statistical tests from the last lab on mitosis, I kind of wanted you to have that done on Monday, though I understand not everybody was able to get it done. So if it's not finished and you're just needing time, then I'd like you to let me know when you think you'll get it done. If you need help because you don't really understand what you're supposed to be doing, then I'd like it if you'd come into my office hours between, well, which is essentially now, but between 11 and 1, and we work through your problem, you know, the questions you have so that you can get that done. But I did want that for Monday, but that's kind of an assignment in my mind that was pre all this digital learning stuff. For Friday, you just have some notes on enzymes that I want completed. And then when we get back from spring break, this lab I just showed you is going to be due on Monday. And then there's another lab, which I'm going to show you guys on Friday that uses that same website that you'll have for Tuesday, although it's not quite so data involved. It's a bit more of just a like, how would I prove this kind of lab?
Okay. And my second question was for like the graphing portion where you were trying to graph the data.
Yeah, failed at it.
Yeah, I was trying to say, so like, like, what were you trying to like represent in the graph? Like, was it?
I'm gonna put another. Yeah, Jermaine, I appreciate it. I'm gonna put another video up here. I'll make it sometime in the next couple hours, and I'll link it in the course in Schoology so that you can see it in the assignment. What I was trying to get was a graph that would have shown reaction rate based upon different temperature variables, right? So your X and Y axis, and you would have seen that the average reaction rate was higher, or well, it was 0 at 0 degrees, and then it was, you know, I don't remember what the values were, 1.9 at 1 degree or at 10 degrees, and then 2.8 at 20 degrees. So it would have gone up and up as the temperature went up, and then it would have dropped. So you would have seen like basically that the enzyme works better as the temperature goes up, but then it stops working after it gets to a certain temperature, meaning it stops carrying out the reaction. It stops breaking down lactose sugar into glucose. And on that graph would have been error bars so you could have said, well, is there actually a difference between the enzyme activity at 10 degrees Celsius and at 20 and 30, right?
Yeah.
Does that make sense?
Yeah, I think so.
Because you could have looked at overlap.
Yeah, okay. So pretty much what we did like prior at the beginning of the year.
Yep.
And stuff.
It's the same kind of statistical tests that we've been using throughout the year. And you know, this statistics and how to use them and how to think about it is just something you just got to keep practicing over and over again. But
yeah, okay.
But again, if you're struggling with that part of it, like understanding what the statistics mean, that's a great thing to hop into my office hours either tomorrow or Friday, and I can help you work through it.
Okay, cool. Thank you.
And I get, like, everybody's got all kinds of crazy things going on that are unique and difficult in their life. So please, if you have questions, communicate with me. If you can't meet during my office hours, just send me an email and we can either find another time we can meet online, or just I'll answer the question online. And if you can't get something done by a due date, if you let me know ahead of time that that's the situation, I understand and fine, as long as we're sort of reasonable about how and when we'll get stuff done. So, all right. Are there other questions? All right. Sounds good. I'm going to sign off then, and I'm going to try to figure out that graph and make a short video of that. And then I will post this video as well as the video of the graph by 1 or 2 this afternoon. So if you want to go in and look at that, or if you know, so, okay. All right, guys. See ya.
Hey, Lindy.
Oh, it's still recording.
Oh, yeah. Okay, bye.